Hi, everyone. Great to be here. Great to have everybody here on this for this terrific Cross-College of Science Colloquium. And thanks so much to Heather Clark for agreeing to give her time and to discuss with us truly the outstanding work that she is doing and her leadership for CILS, the Institute for Chemical Imaging of Living Systems. A real honor and pleasure to have you here, Heather. But let me just say, with my enthusiastic welcome, that's actually all I'm going to say to you. And I'm now going to turn this over the welcome and introduction. And I think you're going to monitor questions as well. Nicole Langlois, tell me.
Yes, Langlois.
Langlois, okay, who is a graduate student in Professor Clark's research group. Nicole, it's a pleasure to turn over to you and to have you introduce Professor Clark.
Great, thank you. So it is my pleasure to be able to introduce today's speaker and my advisor, Professor Heather Clark. Dr. Clark is a professor in the departments of bioengineering and chemistry at Northeastern. In addition, she is the founding director of the Institute for Chemical Imaging of Living Systems and an associate editor at ACH Sensors. She received her PhD in analytical chemistry from the University of Michigan and completed a postdoc in the Center for Cell Analysis and Modeling at the University of Connecticut Health Center. She is a current fellow of the American Institute for Medical and Biological Engineering and has received awards for both research and teaching, including the DARPA Young Faculty Award, and her work has been featured in a live CNN interview, the Wall Street Journal, Wired Magazine, as well as the MIT Technology Review. So with that, I would like to turn the floor over to Heather for her exciting talk today on nanosensors for imaging the chemistry of the body.
Thank you, Nicole, for the introduction, and thank you to Hazel and Aaron for the invitation to be here today. It's truly my pleasure. Let me hide my video here. So today I'm going to discuss one of our projects in the area of nanosensors, and we are working here at CILS to decipher biochemical signaling in the body through imaging. And if time allows, after I talk about a little bit of science, I'm going to tell you about CILS and what we're trying to accomplish. So the need that we're trying to address is to visualize the chemical content of the body. The language of the body is built with chemical signals, so biochemical signaling is very important. We need to understand the identity of these biochemicals, these biomarkers, their locations in the brain and body, and the amplitude and timing of the signal. So we combine nanosensor development and imaging modalities to visualize that chemical signature. And as an example I like to give is that if you see the image on the left, just a standard MRI, and it's a great technique for non-invasively imaging structural detail, maybe even function. But really what's going on as you're listening to me and you're hearing my voice and you're processing that into thoughts and memories. What's really happening is this really rapid signaling event between the neurons, and it's the action potentials that are this cycling of, this intake of sodium, this calcium signaling within the cell, the release of neurotransmitters that start the action potential at the next neuron. This happens really, really quickly. But all of this detail that we know goes on is all lost in this image here on the left. And what we would like to do is add that biochemical signaling component to images such as this. So our goal is to light up the chemistry of the human body. And if you invite me back in a few years, hopefully we'll be moved on to humans. And we're doing this through innovations in sensors, hardware, unique animal models. So our approach is we develop novel probes for biochemical detection. At kind of a basic level, each one of our sensors is composed. If we want to measure a biomarker of interest, we have to have a recognition unit, some sort of small molecule or protein or enzyme that binds to our biomarker of interest. But because we need to be able to visualize this, and most recognition units wouldn't have any sort of way to do that, we pair it with a reporter to tell us that this binding event has happened. And then because we want to work in vivo, we need to hold these two together. And we do this by scaffolds that we develop all the way from polymers to DNA. And on some of our sensors, we add a targeting moiety so it stays in place in the area of interest. We use different imaging modalities in order to address these probes in the body. And it's a real trade-off. So we take a design approach to measuring in the body, and we say, what question are we trying to answer? And what requirements do we have on the system? Do we need really good spatial resolution or really great imaging depth, or somewhere in between? And we choose this imaging modality, and then I always say that I really need to add a back and forth arrow, right? Instead of just a one-way arrow, because this is an iterative process. We choose our imaging modality, we go back and think about sensor design and back and forth in order to really address the question at hand. We then characterize these sensors and imaging modality in animal models with the goal eventually to create a biochemical atlas of signaling in the human body. So why isn't this already done? Why aren't people already imaging the biochemical signaling events in the body? And it's because chemical analysis as we know it, so I'm an analytical chemist at heart, chemical analysis is a trade-off. So if we want to measure a biomarker in the body, one option is to take a sample. So you can take a biopsy or a blood draw, and you can perform chemical analysis ex vivo. And you can do this with an assay or a clinical analyzer, and you can very, very specifically and sensitively identify a specific chemical. But it requires removal from the body. And so really you lose your temporal information. So once you've taken a sample, you don't know what happens 5 minutes later, much less 5 hours later, and which would require taking more and more samples to get some sort of continuous analysis. And that might be okay for a blood draw, but there are, for a biopsy, you certainly don't want to have to take more than one sample. Alternatively, option 2 is you could implant a probe such as a microelectrode or microdialysis in the body. This can also identify specific chemicals and has unmatched temporal resolution, but now you've given up spatial information because you only have one spot in the body which you can monitor in time. Optogenetics is a more recent breakthrough, I guess, in monitoring in the body. These genetically encoded sensors are fluorescent reporters in vivo. You can also identify specific chemicals in the body. They're very cell specific, which is fantastic. You know exactly where your reporters are, but it requires the development of an animal model. And so it's great for mice, maybe not great for other animals. And it's more difficult to tune the wavelength or sensor performance because each one requires its own animal model. And then option 4 is the one that I mentioned before. So imaging in the living body with something like MRI, where you can see functional information. So areas light up, and it's very non-invasive, but it's a surrogate of the actual signal that you want to measure. And so it's very difficult to obtain exact biomarker information with this method. So really what we're trying to do is combine the benefits of option 1, which is, being able to take a sample, with the non-invasive option of imaging of the body, and combine those together so you can get specific information in vivo. The benefits of imaging the biochemical signaling in the body, we feel, the first would be to bridge animal models. One sensor can be used in rodents or C. elegans all the way up to something like the human. And we feel that not only would it be useful to use these probes in any of these models, but you can kind of think of it the other way around too. So if we could measure particular disease levels in the human body, we might be able to have better animal models of that disease, right? Instead of, for something like Alzheimer's or other diseases in which animal models might be difficult right now, we can understand that bridge a little bit better. We could have better real-time trend information, as I mentioned, so you can see levels in the body over the course of days, which would then mean that you could have real-time feedback of therapy response. So if you administer a drug, does the body respond? You would have that real-time information. And of course, We would love to be able to multiplex this and measure several biomarkers at once for a fingerprint of disease. So there are 4 ongoing projects in the lab right now. I'm only going to discuss one today, but I wanted to give a little bit of overview of what the lab does. We currently measure ions in the circulatory system. We measure small molecules, particularly acetylcholine and other neurotransmitters using MRI in the brain. We measure acetylcholine in the peripheral nervous system, and proteins, particularly cytokines, in the immune system using photoacoustics. Today, I have chosen to discuss the peripheral nervous system, in particular small molecules and measuring acetylcholine with fluorescence. I chose it because this is a project we've been working on for several years with James Monahan, who is in COS. So I thought it would highlight nicely our collaboration, our ongoing and long-term collaboration. So a little background into the project itself. So we work with the NIH SPARK program. SPARK stands for Stimulating Peripheral Activity to Relieve Conditions. And the hypothesis of this program is that great physicists and engineers have developed microelectrodes that are extremely good, very high performance, and really have successfully been implanted into the body. So why not use these in order to be an actual therapeutic? That you could implant them onto a nerve that innervates an organ and control the function of the organ with that electrode array. And so, you can think about this for applications really with any organ system, the heart, the bladder. You could regulate bladder function by implanting an electrode on the nerve that makes the bladder respond, right? And then that would really significantly improve the function for individuals with bladder dysfunction. But where we come into it is the actual dose of an electrode implanted on a nerve that innervates your organ. What is that dose? We understand dosing very well for something like a pill that you take, right? You figure out based on a population average, and weight, what dosage you need. But what is that dosage of an electrode array? Do you, set it at 3 volts for 100 pulses and do this every hour or every 5 minutes? Or is it, 50 pulses? I mean, it's the number of choices is almost infinite. But if you could have a real-time readout of the biomarker that is released when you use that electrode to stimulate the nerve, that acetylcholine that's released from that cell and bridges the gap to the organ and triggers its function. If you could measure that release in real time, it would give you instant feedback to how to set your electrode array. And it could be individualized for a particular person who had that implant. So during surgery, you could implant the electrode, you could measure the biomarker that's being released. You could set it at the appropriate levels for that individual. You could then, close up the surgery, and in theory, that then you could then regulate that function. So that's where we come in. We measure acetylcholine, and we can do it very sensitively. So, as just an example here of the constraints that we are working within. We know that acetylcholine is the key neurotransmitter that's released. And so this is an example of 2 cells. Acetylcholine is released from one, bridges the gap between and binds to a receptor, which then triggers the next action potential. Or if it's innervating an organ, it triggers that organ to function. So this space, and the reason I show it to you, is very, very small. This is like 20 to 50 nanometers at best. And so we're really talking about a very, very small area in which we need to make a measurement, which leads us to think we need all the photons we can get. We need a very sensitive imaging technique. We need to use fluorescence for this. We also need some pretty small sensors. So our design requirements, if we're going to pick an imaging system, we need to be able to use a fluorescence microscope. And we're willing in this case to give up invasiveness in order to get our spatial and temporal resolution. So this will be a very invasive technique. And so we need to think about the size of our sensor, which we'll go through on the next slides, the dynamic range that we're working within, the sensitivity. And by sensitivity, I mean the making a small measurement on kind of a large background. So quite often when you're thinking about diagnostics, you say, I'm trying to, measure a needle in a haystack. But in this case, what we're doing is measuring a few extra pieces of hay that are added or subtracted from that haystack. So we're making a measurement of the addition or subtraction of more acetylcholine on a background, a potential background of acetylcholine that's already there. The system needs to be reversible so that we can measure continuously. And then very importantly, we need to deliver this to the body, target it to the area of interest, minimize the background labeling of our probes, and then image through tissue. And we're going to show this all in an animal model. So we turned, because of the small size of the system, we turned to DNA nanotechnology as our scaffold. So we have that recognition element, the reporter, and the scaffold. And we use DNA for that scaffold because so much advances have been made by other researchers in programmable design. So DNA origami. You can design a template strand and pair it with staple strands, and then anneal it in solution where it will self-assemble into really basically any shape or size that you want. You can kind of see the video of this assembly. And other labs have made, really cute structures, little robots and smiley faces and so on. Our design, which you'll see on the next slide, is nowhere near this, this adorable, I guess, but it's really for us, this programmable nanostructure has many, many advantages. One being that every single one of our sensors is exactly the same shape and size as every other sensor, which is hard to do with something like polymers. And we can also put a lot of chemical functionality on the scaffold itself so that we can snap into place with click chemistry, our reporters and our recognition elements and our targeting moieties. And we can do this with really high yield of ligand attachment. So, this video. Oops. I forgot that I have trouble playing this video. So our sensors, in short, I'll show you on the next slides. We attach our reporter and our recognition element to separate strands of our DNA, anneal them together in solution. And then when a acetylcholine molecule is cycled by the enzyme, it leads to a change, which is then reported on with our fluorescent reporter. So specifically, our design looks like this. If you can see here with my little arrow, we have this dendron structure with different branches. This scaffold is made out of the DNA, in which we attach an enzyme, acetylcholine esterase, and a reporter of that function, in particular, a fluorescent pH indicator. And the way that this works is that the enzyme binds to and degrades acetylcholine. It creates an extremely localized acidic environment around that enzyme. So the DNA allows us to attach that reporter very close to that enzyme molecule so that it will report that localized change in pH. And it will modulate the intensity of its signal depending on how much acetylcholine is being degraded in the surrounding area. We also add bungarotoxin to the other end here of that scaffold in order to target it and bind to the acetylcholine receptors at the cell. And then finally, we put on an internal standard. This internal standard is a fluorescent reporter that does not modulate its fluorescence intensity. And so it basically gives us always an idea of how many sensors are in an area, right? So because we can't control that, all we can do is add a solution of sensors. But by knowing exactly how many have bound to our receptor, we always have that internal standard so we can make a quantitative measurement of acetylcholine. Our nanosensors are fabricated from the bottom up. So we take our DNA strands and attach our fluorescent reporter to some of the strands. We can then anneal it in solution to get our scaffold that has the 4 fluorescent reporters, and then functionality so we can do the protein attachment steps. We don't add the protein before annealing because it would denature during the annealing process. And then the proteins are attached through just standard click chemistry. So we use a maleimide linker to add the acetylcholine esterase, and we add a TCO linker to attach the BTX. And both of these result in stable attachments to the scaffold. We can then purify these through size-exclusion chromatography. And if you had gone to Nicole's talk at ACS today, she has a recent paper in analytical methods out on how to use size-exclusion chromatography efficiently for DNA origami. But size-exclusion chromatography, in short, separates particles by size with the large eluting first. And because our sensor has so many fluorescent reporters and proteins attached to them, we can see exactly where the full sensor with all of the attachments elutes from the column, and we can isolate those. And then by gel, we have validated that this works. So I could show you some more analytical characterization, but I will actually just jump ahead a little bit to showing how this works in vivo. So in theory, this should be a great sensor to monitor acetylcholine, really basically anywhere in the body acetylcholine exists. And since we're interested in the peripheral nervous system, we wanted to validate the technology in an organ system. We chose the submandibular ganglia, which is actually the system that stimulates the saliva glands. And we chose it not because it's the most exciting organ in the body, but because it is fairly close to the outside of the animal. So it's located here on the sides of your neck. And so this kind of outside location still is too deep within the body to just use fluorescence without doing surgery to expose the SMG. But at least it's a much more simple and straightforward surgery because it's not too deep within the body cavity. And so this is a great system, or we thought it was a great system in order to validate this technology for use in the system, because no matter how much in vitro or on the benchtop validation one does, you learn so much by going in vivo, and you really need to do a full set of analytical characterization to make sure the technology is functioning exactly the way you think it is. So my arrows have shifted here, but if they hadn't, you would see that you can see these nerves here and the gland. And these little bumps on the sides of the nerves are the SMG. And these are the targets that we're trying to image. So you can imagine in a mouse how incredibly small this is here. And this is just a fluorescence image. This is a GFP labeled SMG. You can see how small they are, and we can place an electrode across the nerve to stimulate that SMG, which would then stimulate saliva production. So the next slide is a little bit more graphic. We can use any sort of fluorescence imaging technique. We're not limited at all. If it fluoresces and you can capture it with a microscope, it works. And so we use a very, very basic system, actually. This is just a standard upright microscope. We have a light source, and we position the objective above the SMG. We can excite the fluorescence with the light source and then capture that fluorescence emission back through the objective. We then put a beam splitter on the camera so that side by side, simultaneously, we're taking a picture of the SMG of both the reporter and the internal standard. So we have, we can basically ratio those two and get a quantitative measurement of acetylcholine being released. The kind of tricky part of this is that the SMG is still there in a lot of connective tissue and fat in the system. So what we did was we opened up a cavity in the mouse and held it open, filled it with solution, and then took a very small spatula and slipped it under the SMG and pulled it just a little bit out of that cavity so that it was taut. And this gave us two advantages. One, it made it easier to image with that microscope objective, right? It just holds it there at a place where it's easy to address with the objective. But secondly, it also pulls it out of the body a little bit to get away from movement artifacts from breathing and heartbeat. And it's not perfect, but it certainly helps to stabilize it in that way. And if you remove an SMG from the body and mount it and look at it with a much fancier microscope than what I'm going to show you today in the live images. So this is confocal, and the green circles are cells that have GFP loading, and the red rings around it, or the orangey rings around it, are where sensors attach at the receptor. And so you can see that the sensors attach in kind of a ring around the cell membrane of each one of the cells that makes up the SMG. Delivery is always the trickiest part. This was, when we envisioned using this, we envisioned a solution of our sensors that we could just drop on and start doing imaging. It's never as easy as we think about when we're putting together the kind of plan. And what happened was that even though we had taken this system and put the spatula under and removed a lot of the connective tissue, Michelle found that although she was extremely talented with doing this surgery, there's no way to remove every little bit of connective tissue across that nerve system. So she had to take a pulled glass needle and come in at a 25-degree angle and add just a little puff of solution of sensors just to get underneath that connective tissue. And if you look here on the far right, this is the SMG delineated by the white lines. And she has a needle in there, and there she has puffed in a solution of those sensors. And what you see is obviously the fluorescence. You can see the fluorescence from the sensors right away. But also you can see that how quickly the sensors attach to the receptors, and that the sensors that are not attached are washed away. So we get rid of that background. The diffusion is pretty quick through that system. So once we had a delivery system in place and we could efficiently label the tissue at the SMG, we then performed a secondary analytical characterization. We prefer to do this in vivo, but because acetylcholine is a stimulant, what we had to do was go through all that labeling process and then post-mortem remove the SMG from the animal and mount it underneath a harp. So you can see the gland here and the SMG underneath the harp, and then put it under the microscope and add acetylcholine in solution at various levels. So we control the amount of acetylcholine, and as you add more acetylcholine, the more it lights up, which is exactly the way you would expect. And then we can analyze this data, plot concentration of acetylcholine we added versus the fluorescence, the rate of fluorescence intensity, build a calibration curve. And with this calibration curve, we can find the lower limit of detection. So the area where we still feel the lowest amount of acetylcholine, where we feel comfortable making a measurement, which was 228 nanomolar. But we can also, in our next in vivo experiments, use this calibration curve. So when we're doing our in vivo experiment, we say, oh, if we measure 200, we can use our calibration curve to say that concentration we measured was, whatever the level is. So actual in vivo data looks like this. So this is the system. The animal is alive. We have mounted electrodes on the nerves to stimulate the SMG. We have analyzed area of region of interest, just one area of interest outside of one cell in the SMG. We stimulate the nerve with the electrode at 3 volts, 10 hertz, 10 millisecond pulse duration for 100 pulses. The blue line is the average of 3 separate animals. And we start the stimulation here at the arrow. You can see that at stimulation, the acetylcholine rises fairly quickly with a slower clearance from the tissue itself. Secondarily, we have run a control. This control is the same surgical procedure, and it is the same addition of a sensor. But the sensor is a little different. So it has the same scaffold and the same targeting and the same reporter, but we have not added the enzyme, that recognition element for acetylcholine. So all it does is measure pH. And this is a great control for our sensor itself, obviously, but also for the system. And to make us feel confident that what we're measuring in our real system in the blue is actually acetylcholine that's being released, and that we are not just so overstimulating the system that we're changing the pH globally of the biological system. And so you can see here in the green that at these stimulation parameters, there is no global change in pH. And so we can feel confident that here in the blue, it's actually acetylcholine that we're measuring. This is dose dependent, which is good because that's what the rest of the Spark team needs, right? So if they're going to use this in order to monitor the release of acetylcholine at the system, they need to understand whether or not it responds to different levels. And so this dose dependence is both by changing the voltage. So if you change it from 10 volts to 5 volts to 3 volts, you see the overall intensity go down, and the number of pulses that you use at 1 voltage. So if you hold it at 3 volts, but change it from 100 pulses to 50 to 10, you can see that this dependence on the number of pulses as well. And then of course, if we stimulate and then let the acetylcholine clear from the system and then re-stimulate and re-stimulate, it's a reversible process. So you can see this in real time through repeated stimulations. And it's quantitative. So as I mentioned before, we can use that calibration curve. So if we choose a region of interest, which would look like this over time, and then use our calibration curve to make a measurement, we can go through and find that at this particular region of interest, the amount of acetylcholine that was measured was 2.7 micromolar. And you can analyze any of the regions of interest in that system. And then finally, to highlight how important it is, or maybe some of the advantages to being able to image rather than just measure one single area, to have that spatial information. When I had envisioned this experiment, I had thought that what we would see is that when we stimulated that nerve, we would see acetylcholine kind of burst from across that entire tissue upon stimulation. But we worked with Kira Pascanzer at UCSF, who had developed a system for measuring calcium imaging in astrocytes. And so we modified her Aqua program to map the release of acetylcholine across the SMG. And what we found was not that it burst from kind of globally across this tissue, but instead the release began at the top of the SMG and then propagated across that tissue itself. And you can see that propagation pattern in the movie. When it starts over again, you'll see. And so maybe in the SMG, this isn't, so you can see it quite nicely propagate across that tissue. And so maybe in the SMG, this isn't important biologically, but as we move forward and we start imaging in the heart, which is what we're doing now, I think this will be really important to have this spatial information from imaging. So in summary, before I talk about CILS a little bit more globally, I want to highlight a few future directions. As I mentioned before, we've started working in the SA node in the mouse heart. We're developing a sensor based on the same platform to be able to measure norepinephrine, because that's also important in the heart. We're also making a modification in the targeting moiety to target the receptors in the heart so that we can do two neurotransmitter imaging simultaneously. And then possibly making it a little bit smaller so that we can get more into the synapse rather than kind of a global release from the area around it. So before I switch gears and talk about CILS really quickly, if I have a few minutes, really want to thank the team. I highlighted the postdocs and grad students that contributed to this project as I moved along in the presentation, and also want to give a warm thank you to James Monahan, who has been such an important part of this whole process. So with that, I'd like to discuss CILS just a little bit and what we're trying to do here on campus to think about imaging the biochemistry of the body in a way that goes beyond what maybe two collaborators can do on one project. How can we really move this forward so that, I don't really know where my words went here, but so that we can really get to this goal of thinking about how do we image in the human body. And so I really think that this is a very hard problem, obviously. So we need great probes, which I just discussed, earlier, but we also need advances in hardware for imaging. We need unique animal models like what James Monahan does with the axolotls. What can we do with a tissue that's completely transparent, right? I mean, it's really could do much faster, quicker imaging, right? And then we have to bring in a knowledge that exists in areas such as drug delivery, right? So people who do drug delivery think a lot about how to get drugs to certain areas of the body. Well, we think a lot about how do we get imaging agents to different areas of the body. So if we bring those people together and tackle these tough problems and say, okay, now what if our design requirements, we're really, we think about what we need, but then we can mix and match different animals and probes and imaging hardware and drug delivery knowledge so that we can address any problem, right? We can think beyond even the peripheral nervous system or the one biomarker and really build this atlas in the body. And so we've brought together expertise from across COS and COE. We have engineers and biologists and physicists all working together to address these problems. And our plan in CILS is really to think about how do we move beyond, two collaborators writing an NIH grant. So we're working to build a strategic plan, which is building partnerships so that we can really diversify our funding portfolios and work with collaborators and commercial funders so that we can move forward in different ways. We want to be able to validate and disseminate this technology, really move beyond writing a paper. I love that paper that I just presented to you today, but really where our impact will be is when somebody who has a biological or a clinical problem uses that technology to answer it, without me even being a collaborator on the paper, right? So what we want is somebody to just be able to use these tools and really unlock questions that they couldn't unlock otherwise. I mean, we look at the field of calcium imaging, and with the first invention of fluorophores to be able to measure calcium. And then it just changed everything about the way we think about the body and how we can make a measurement. What if you could measure anything, right? And what if anybody could do it? So we're really working to try to get these tools out of our lab. We have a core facility at the core of all of this. And so we've built a partnership with Zeiss We have a gorgeous core facility in the basement of ACh, which I'll show you a picture of for everybody at Northeastern and really beyond in the greater Boston area to be able to use this state-of-the-art imaging facility. We want to give the support to our CILS members to be able to, take these high value projects and move them forward with all of these different components, and also to increase our outreach so that the future generation of scientists are excited about imaging as well. So a few of the things that we're working on right now, we're working on a program incubator, which we call the CILS Accelerator. This is led by our Executive Director, Jean Civilico. So if somebody in the CILS program has a really cool technology and we think it has commercial impact, so what we're trying to do is start an accelerator where we can nurture that project and work. So Jean can work to, first of all, link it to potential commercial investors, but nurture it so that it's to a maturity stage where those commercial investors are ready to take it forward. Many of our projects in academic labs result in really great papers, but that's still a very early stage for commercialization. So we're looking to nurture those programs with SBIR funding, STTR funding, so that they're ready and aligned with commercial partners. And for our outreach, we have partnered with BioBus. And BioBus has been very well established in New York City. It is a system of buses which have been retrofitted with microscopes. And they drive to K through 12 schools, and they're like a field trip that comes to the school. And they park outside the school. Graduate students and staff members work with the K through 12 students to introduce them to microscopes and what they can do and really give them a hands-on experience. They were interested in moving to the Boston area, but if you think about the logistics of driving a bus around Boston, there's no parking outside of the schools in the greater Boston area, and it's very hard to get down some of the streets. So we have teamed with them and provided a small amount of space in the CILS core facility, in which we've partnered with Dase, again, who have donated 8 microscopes for the surrounding school systems to be able to come to Northeastern. We're going to have grad students involved with CILS, ready and trained to work with the students. And so that although the field trip can't come to them, at least it's a very short trip to come and see these facilities and really get a hands-on microscopy experience. And then finally, we have a gorgeous core facility in the basement of ACh. It is open to anybody to come use. I really, really recommend. This image here was of brain prep from Rebecca Shansky's lab. And look at the way it comes alive on the light sheet microscope. It is just an absolute gorgeous image. This is a whole tissue section, and you can see in just exquisite detail the structure of this sample. We have confocals and 2-photon light sheet. We have a small animal MRI, and we have a cell sorter and flow cytometer. And so with that, I think that the future of CILS and Northeastern is not just CILS alone. We're not just bringing together the faculty that are in our particular institute, but, really to be able to integrate with other institutes and other regional campuses so that we can, take an even bigger approach to answering some of these fundamental questions. So with that, I'd like to thank you for your time and answer any questions that you might have.